I'm just going to draw ones on the receptors, or I'm sorry, on the membranes of cells. So that's a cell. And I'm going to put a receptor in that membrane. And I'm going to draw it a specific way. I'm going to make it so that it has that kind of shape to it. Okay? Like a little triangle would fit in. So that is a receptor. Now, what is the job of that receptor? The job of a receptor is to bind a molecule, a very specific molecule. And we're going to draw that molecule. So we're going to call it receptor A. I'm going to call this molecule A. Doesn't matter what the molecule is. And you're going to draw it, you should draw it with like a triangle. We want it to fit in there like two pieces of a puzzle. We want it to be specific for that particular receptor. And that is a word that you see in your notes, specificity. Specificity is talking about how we have something, a molecule in this particular case, and a receptor that are specific for each other. And it's based on structure. And so that molecule, A, will fit in there like a little puzzle piece. Now what? Well, the receptor has started to do its job. And again, its job is to bind something. And once it does bind whatever that is, that specific molecule, and we'll call this specificity. Again, it's in your notes to find. We're just kind of drawing a picture of things. Something happens inside the cell. Now, right now, I'm going to call it, so we're going to say the very first step is that. The first step is the receptor binds the molecule. That's step number one. The second step, this is what literally I'm going to call it now. Stuff. Stuff happens inside of that cell. What kind of stuff? Stuff we'll talk about at a later time, and a lot of stuff that's underneath what you see in your notes that you're not going to have to know for right now. But I promise you that I'll be talking about stuff at a later time. In essence, this receptor is telling that cell what to do. But that receptor cannot tell that cell what to do unless it is first activated by some molecule that binds to it. It needs to be turned on. What turns the receptor on? A molecule binds to it. Okay? That's pretty much what we have to know up until this point. So what did we draw? We just drew that. That's what we just drew. Okay? And for right now, that's all you have to know. There's way more on receptors later on this semester and next, I promise. Okay. All right? Now, a couple of other terms that we need to be familiar with. Agonist and antagonist, activator, blocker. So agonist and activator, same thing. Antagonist, blocker, same thing. Now they're defined here in your notes. What is an agonist? What is an activator? What it does is that it activates a receptor. What an antagonist does is it blocks the receptor. It doesn't allow the receptor to do what it wants to do. All right? I'm going to give you a specific example. Later on this semester, I'll give you many specific examples. But right now, I'll just give you one. Okay? So let's talk about an agonist. Let's talk about an antagonist. We're going to say, you know what? We're just going to stay on this page. We're going to say that receptor A, when it is activated, and we know how it's activated, because molecule A is what's going to activate it when it literally binds to it. Receptor A, when activated, causes the cell to increase, we'll make an arrow, your blood pressure. There are cells in your body that do this, by the way. We'll talk about it next semester in excruciating detail. But we're just going to do something very simple right now. Okay, so there are some receptors in your body that when they're activated cause your blood pressure to go up. There are some receptors that will cause your blood pressure to go down. How that happens, I'll tell you next semester. Okay? Now let's talk about agonists and antagonists. So, an antagonist, I ran out of room, maybe you didn't. Antagonist to, again, in this particular example, receptor A. What's going to happen? Now, what would the antagonist be? It could be some drug that you give somebody. Okay? It could be something else in the body that can interact with that receptor in a way that doesn't allow it to happen, that stops stuff from happening. what would be the result of this? An antagonist to receptor A will do the opposite of what receptor A wants to do. An antagonist to receptor A, given the specific example I've given you, would decrease your blood pressure. Anybody over here beta blockers?
Yeah.
Alpha blockers?
Yeah.
Okay. ACE inhibitors?
Yeah.
Those things lower blood pressure because they block stuff. They block receptors within your body. Given what I just said, then an agonist to receptor A would cause what to happen to blood pressure? It would go up even more. So that would increase your blood pressure. It would activate that receptor even more. There are drugs you will give people that do raise blood pressure because blood pressure is too low. There is circulatory shock, for example. We'll talk about it next semester. All right, so I've given you a specific example. But regardless of what example you see on the exam, which is when? A week from today or Tuesday, depending on when you have lab, because your exams, your physics exams are in lab, correct? Is there a lecture here Monday night? Yes, there is. There's a lecture every single time we're scheduled for lecture. Even if there's an exam, even if there's a quiz. As you guys know, because half of you or more have a quiz after this lecture, right? Okay, try to give me your attention if you can, because this is going to be important stuff. That's all we have to know for that. So if on the exam, I give you an example of a receptor that does something specific. If it's blocked, the opposite is going to happen. Okay? If it's activated, more of what it does is going to happen. Okay? Just as I showed you. End of this chapter. All right? Much more on this kind of stuff later. Now what? Change gears. Last chapter for the material that's going to be on our first exam. First exam, first three chapters, as you know, because it's in the syllabus. Let's talk about membrane transport. If we had to draw the big picture of this particular chapter, this is what our picture would look like. So I'm going to draw a cell here. I'm going to draw a couple of tubes. There's a tube. There's a tube. I'll say that's a blood vessel. The fluid in that is blood, but specifically the liquid part is plasma, right? We know that. What tube would this be? The hell am I doing? I just wrote it down before I even let you answer. Lymph. What would the space be that surrounds the lymph vessel, that surrounds the blood vessel, that surrounds the cell? What's that space called? Interstitial. Right? Again, because we learned this, right, that first lecture, I can freely say these particular terms because we know what they are. So we have the interstitial space here. These are all our compartments that we talked about, minus transcellular, correct? So we have three extracellular compartments here, blood vessel, lymph vessel, interstitial space, interstitial compartment, and we have the intracellular compartment, the one within our cells. Again, this is a picture we've already drawn. Now, when it comes to transport, this is our big picture for this entire chapter, is right here. We're going to see how things are transported in and out of these compartments. So this little dot is going to be a solute. That solute can be a protein, an amino acid, chloride, sodium, potassium, hydrogen, doesn't matter. We're going to see how solutes go across membranes, how solutes go from inside of the cell to outside of the cell, how solutes go from the interstitial space inside of the cell. We're going to see how those same solutes can move into the blood vessels, how they can move out of blood vessels, how those solutes move into lymph, out of the lymph into the interstitial space. We're going to see the movement of water into the lymph, into cells, into blood vessels. Okay? Out of blood vessels, we're going to see the water leaving. I'm going to give you specific examples of that, how water is going to be leaving. And then there's another term that we need to be familiar with that we'll talk about on Wednesday, but I'll give it to you right now. Not water, but fluid. And so I'm going to write the word fluid here. How fluid goes into cells. How fluid leaves cells. How fluid enters the lymph, how fluid leaves the lymph. Same thing with blood. I know it's ugly and messy, it's okay, you're not going to see this on the exam. I'm giving you the big picture of what we're going to see. We're going to see how stuff moves in and out of these compartments. That's what we're going to do. We're going to talk about how solutes do it, water does it, and fluids do it. That's what we're going to do in this particular chapter. I'm going to teach you specific mechanisms by which this occurs. There's the big picture of this chapter, right there in a nutshell. All right? Now, when it comes to and this is always happening, by the way, this never, ever, ever stops. Well, it's going to stop someday. All right? But when we're still alive, this is working. And if this does not work properly, what do we lose? What do we lose? Starts with an H. Homeostasis. This is part of homeostasis, guys. Fluid moving in and out just the right way. Water moving in and out of these compartments just the right way. Solutes, same thing. These things have to be moved in a controlled manner in order for homeostasis to be maintained. All right? And we're going to see these types of things over the next 2 semesters. So, when it comes to the movement in and out of all of these compartments, there are 2 ways it occurs. So there are 2 main transport mechanisms. Number 1, passive. Number 2, active. Now I'll just give you the punchline right now and tell you the big difference between the 2. Active transport mechanisms require energy in the form of what? I heard it, ATP. Active transport mechanisms, we're not going to get to them tonight. We're not even going to finish the passive mechanisms tonight. Those require ATP. These do not require ATP, so they don't use them. That's the big difference between the 2. But regardless of what mechanism we're talking about, it's the mechanism is still going to move something from point A to point B. All right, from one compartment to the next. Okay? So there are 4 passive mechanisms that we're going to discuss. There are 5 active mechanisms that we're going to discuss. Some of them we're going to go through very quickly. Others, like osmosis, believe it or not, we're going to spend the most time on. Okay? We're going to just get done with osmosis today. I hope. All right? So, this right here is another big picture of what's going on. So what do we have here in this previous picture? What we have here is just a little piece of a membrane. By the way, what's this called, the blue stuff? Phospholipid bilayer, right? It's phospholipid, it's 2-layered. This is the outside of the cell. This is the inside of the cell. So this is the interstitial space. This is the intracellular space. Now we have these different colored circles here. Those are solutes. And we're seeing in each case, except for one, these solutes are being transported across the membrane. That's what we have. Now, when it comes to our membranes of cells, of organelles, by the way, things move in and out of organelles. Just not in and out of these compartments, but actually in and out of organelles. We're just going to keep it with our compartments for right now and forget about the organelles for now. When we have the movement of these molecules through the membrane, whether it's in or out of the cell, in this particular case, they chose to just move these molecules into the cell. They could be moving out, it just depends. The membrane is said to be permeable. When you see the word permeability, I want you to think of a word that rhymes with it. So permeability, I want you to immediately think of the word ability. If a molecule is permeable, whether it is water, whether it is a fluid, whether it is a solute, it means it's moving from point A to point B. It means it's going from one compartment to the next. So a molecule that is permeable is being transported. Okay? So a molecule, it doesn't matter what it is, molecule that is being transported across the membrane. There's not 2 R's in that. Across a membrane. Doesn't matter what membrane it is permeable.